This is HealthCall Online, the place for extended versions of interviews you hear in our weekly radio broadcast, the HealthCall Radio Hour, now heard across the country in national syndication. Our focus again today is your amazing human brain. And one of the most amazing things about our brain is what it can store and remember. And we'll learn a little bit more about what happens when your memory fails and why is it that you can remember some things but struggle so hard for others. We're going to jump out to Cedars-Sinai and the lab of Dr. Ueli Rutishauser. He's a PhD, has a very impressive resume that I'll link to in the show notes, but we'll just suffice it to say he's a professor of neurosurgery, neurology, and biomedical sciences at Cedars-Sinai and the founder of the Rutishauser Lab. So glad you're here. Help us understand just what is memory? Does it work like a computer stores data on a hard drive?
Thank you, Lee, for having me. It's great to be here. And it's great to talk about one of the most amazing feats that the human brain achieves, which is, as you point out, memory. So you're asking whether memory in our brain works like memory in our computer. So in our computer, memory is stored in dedicated chips. They have RAM. They are called. So those chips, the only thing they do is they store memories permanently. And that's very much not the case in the brain. The human brain stores memory in a very different way. It stores memory intertwined in the actual neuronal hardware, so to speak, that allows us to think and to compute. And the very same circuits modify themselves to also store memory. So it's a very different substrate, and that substrate does not allow a random access like you have to a computer memory in your phone or in your computer.
So is it an electrical signal that is stored? Is it a chemical signal that is stored? What exactly, how does our brain store all that data?
Yeah, so ultimately to make a memory in the brain, you have to modify it permanently. And how that happens is synapses are modified. So synapses are made of proteins. So ultimately, when you make a new memory, proteins have to be produced and inserted and moved into your synapses. So it's an actual physical change.
Protein-based physical change within your brain. So is there something about the intensity of the experience that embeds it more deeply? So, for example, if it's something that is extremely emotionally intense, am I more likely to remember that as opposed to something that's less intense?
Yeah, so to understand that, we should step back a little bit to think about how we think. So ultimately, when we think or make a decision or look at something or have an experience or have a phone call, those experiences are electrical. So neurons generate electrical activity and they communicate with each other by passing that electricity indirectly to each other. So what we need to find out is how electrical activity, which is transient and goes away, makes it into something that's permanent, which is an actual structural change, the so-called engram, as we call it, which is the physical substrate of memory. And so very much so, if an experience is more intense, it's more likely to be stored for the long term in a memory. So there are many factors that determine whether a memory will stick or not, and whether it will be vivid or not, or it will be detailed. And one of them is, as you point out, emotional intensity, absolutely. Although not too intense, because it's, as you may have experienced yourself, some memories, some experiences you may not remember at all if they were very intense or maybe traumatic.
Yeah, so your brain is trying to, I'm assuming that what's happening there is it's trying to protect us from reliving that trauma.
Yeah, so it turns out that there are some experiences that we don't remember, and we think that one of the reasons for that is because at the time that experience happened, there were simply more important things to do than to make a memory.
Okay, I get it. I'm running away from the tiger, and that's more important than remembering where I was when the tiger.
Exactly. That's really secondary at that time from
Yeah.
an evolutionary point of view.
Yeah, I get it. Okay, that makes a lot of sense. So what goes wrong when we can't remember something?
Yeah, so there's two fundamental reasons why you may not remember something. One, the first is it may not have been stored to begin with. So the memory was never made. And there are many reasons for that, but one of the main reasons that we know about is that the brain has what's called a novelty gate. So if something is perceived as being new, novel, then we haven't seen it before, so then we should remember it. But if perhaps by mistake we already know something when we actually don't, then it's less likely a memory will be formed because we think we already know it. So that's what's called an encoding error. It's not encoded. And then the other principal reason of why you may not be able to retrieve a memory or have a memory is it was encoded, but you're not able to find it or retrieve it. And that points to the fact that our memory is organized in what's called associative. We link things to each other. So we can remember if I ask you, well, hey, what did you eat for breakfast last Sunday? You may think, well, okay, I don't know, but okay, where was I and who was I meeting? And you can sort of jump the links and ultimately you may get to it or you may not. But it's often possible for us to declare accurately that a memory exists, but we are not able to get there at the moment because it requires this sort of elaborate search to get to it.
So that newness factor might explain why memories associated with travel and trips and going different places are often a little more easy to recall.
Absolutely, yes. So one of the strongest modulator of memory formation is indeed novelty. So if you go some place where everything is new and you have never seen it, you will make more memories and stronger memories. That's very much true. And that will also very strongly influence your perception of time, because one way by which we perceive time is by how much new happens.
Oh, that's interesting. So if I want to put myself in a state of high memory capture, if I'm trying to learn something new, studying for a test or something of that nature, can I apply that ability somehow? What does that tell me about creating more durable memories?
Yes, so you will if you have it often helps to, when you study something, to not just restudy exactly the same thing, because that would already be familiar. It would be strengthening existing memories, but somehow to create new memories by adding something new about it, to looking at the same situation from a new point of view, let's say, or from a new case, or by adding some new information, and that will re-enable the brain to form memories, or perhaps do it in a novel context.
Yeah. So does that mean going someplace new to study? Does that help, or it doesn't work that way? It's the content of the information I'm trying to retain that has to be new, not the environment in which I studied it.
No, environmental novelty is also very effective, although you're then counterbalancing with another effect, which is it is often easier to retrieve a memory in the same context in which you first experienced it. So there are reliable effects, such as when you study for a test in a given room, or just as there are many memory experiments. They're so-called the doorway effect, and you move the subject to another room, and we will have them retrieve the information, and they will do worse because the context changed.
Oh.
So there are many different effects here. So context changes are not, they make stronger memories, but they may also make it harder to retrieve.
So let me take a leap here. If I am concerned about memory loss and I feel like my memory is slipping, is it help for me to seek out new experiences and get lots of new stimulation? Is that going to help me recover all memories, or only those that are recently formed in that new environment?
I would say in general that's highly beneficial, because we know novelty has a strong effect on the brain, also in terms of emotion, in terms of our arousal. Novelty leads to release of of dopamine, which modulates or strengthens neuroplasticity. So in general, I think that would be very helpful in such a situation, because it will engage your memory system. It will provide new experiences, which will result in new memory, and that, strictly seen, will only affect those new memories that you are creating. But I think the overall arousal that it will provide is going to be very helpful. I would definitely recommend that.
Okay, so then let's flip that around. Does that help explain why we see the elderly decline at accelerated pace once they're in a nursing home or memory care center, where it always looks the same, everything's the same, it's routine? Does that degrades our ability to remember?
I would say so, yes. In fact, there are good studies that show that we can build up a cognitive reserve, and remaining cognitively challenged and active and perhaps uncomfortable sometimes really helps healthy aging. And if something is always the same every day, we don't do anything new, we are not challenged, that's detrimental for brain health. There's no doubt about it. The brain is not a muscle, but you can view it as a muscle. If you don't use it and challenge it, it will degrade. So I would think that's really highly detrimental. Yes.
So tell me about what determines when a short-term memory gets converted to long-term. Do I make that? I don't make a conscious decision about that. It kind of just happens, doesn't it?
Yes, it does. So that's a very interesting question. So the short-term memories are memories such as, I'm telling you a phone number, and you walk downstairs to dial it, and sometimes you arrived and you're like,
what?
Why did I go downstairs?
I really want to remember this. I'm going to remember this number. I'm really going to remember this, and then you don't, right? I mean,
exactly.
That happens.
So you're pointing out it's really effortful to keep a short-term memory, and the reason for that is because short-term memories are not based on physical changes in the brain. They are based on ongoing electrical activity, and it's very easy to disrupt that activity or to let it die down, and then it's gone. Whereas, as we discussed, the long-term memories, they're based on physical changes. So the question is, which of these activity patterns that are going on during short-term memory actually make it into a long-term memory? And very few do. For example, you probably remember you have probably stayed in many hotels in your life, but I don't think you remember which room you stayed in for many of those days. But while you were there, you remember that, right?
Yeah, I'm in 220 or whatever, exactly, yeah.
Exactly, but then once you're gone, it's basically now doesn't stick in memory. So how we prioritize things that make it into long-term memory is a fascinating question, and one of them is whether it's linked to a prior relevant memory. Relevance to our life, and novelty also. Whether it was high, like if that room was extremely unusual, you may remember that number, right? Or if something special happened there. But otherwise, probably not.
So what is the connection between smell and memory? You hear people say that smells trigger a memory so powerfully. What happens there?
Yeah, so smells, just like any other senses, they can be associated with memory. So they can be one of the links that allows you to reach one of the memories. So smells, there's two main reasons why smells are so powerful. One of which is when this happens, usually the smell is novel. It's something you've never smelled before, like it's some kind of unusual smell. So it's novel, it triggers the novelty system. And the other is that the sense of smell has prioritized access to the brain. It's the only sense which reaches the cortex, the most high-level area of our brain, the neocortex, directly. It bypasses one of the relay stations, which is called the thalamus. And the other senses, vision, audition, touch, they have to go through that additional relay station. So it's thought that the reason for that is because the smell is the sort of the most archaic sense, the earliest in evolutionary times.
Interesting. So Dr. Michael Roizen, a pretty noted gentleman who studies longevity factors, told me that if you seek out and smell 4 new smells a day, that helps support your memory. Does that ring true with you? Does that make sense?
Well, from a novelty point of view, for sure, because you will then have 4 novel, experiences a day that you can link things to. So I would say so, yes. Although I would think it'd probably be very hard to find 4 new odors every day, because one of the interesting things about odors is it's fairly low dimensional. There are really not that many distinct smells.
Yeah.
Compared to, images, we can generate as many as we want. But I would agree with that. I think that could be helpful from this novelty point of view. Yes.
To give your brain a little bit of exercise. Okay.
Yes.
So I'm interested in learning more about the single neuron studies that you do. So you're actually looking at the cells forming a memory. Is that right?
Yes. So let me talk a little bit about how we do this work. So how do we actually know some of the things that I talked about? So one of the reasons why we know little about the human brain and its diseases and how to treat them is because it's very difficult to study it for obvious ethical reasons. We can't do many things that we would need to do to really figure out how it works. And one of those is that we need to be able to listen to the electrical activity of individual brain cells. So individual brain cells fire action potentials. We call them spikes. So we need to know when individual brain cells spike and when others don't. And we need to be able to do that while somebody learns, to see how the activity of given cells change as a function of their experience. So most of what we know about how the brain achieves this is from doing this in animals, with animal experiments. That's how most knowledge has been developed in most treatments. But some of these things we just cannot study in animals. It's very difficult to ask an animal, well, what do you remember about yesterday? So to our luck, there are some clinical treatments that require inserting little wires into the brain of human subjects. And these wires can be used to perform such recordings. And of course, we can't insert that just into a volunteer, but there are some clinical situations where this has to be done anyway for the treatment of a disease.
Oh, like epilepsy or Parkinson's with deep brain stimulation, that type of thing?
Exactly, those are the 3. So in those scenarios, we can, without adding any risk to the procedure, we can ask the patients who come here to the hospital to be treated, we can ask them whether they would be willing to participate in a research study while they're undergoing their clinical treatment. And in those scenarios, we can do memory tests and listen to their neurons without modifying the risk of that procedure, which is substantial, but it's not related to the research.
Yeah, I get it. So what have you learned about that in watching the neurons respond?
So what we have done is we have focused our work on an area of the brain which is called the hippocampus. So the hippocampus is a big part of the human brain which is difficult to reach. It's sort of deep down in the middle of the brain. And what we know about the hippocampus is that it's really essential for making memories. So if for some reason your hippocampus is not functional, you're not able to make new memories. Coincidentally, the hippocampus is also one of the brain areas that often generates epileptic seizures. So those wires do land there, so we can study it. And so we have isolated specific types of neurons in the human hippocampus, and one of them we have come to call novelty neurons. So these neurons react when I show a bunch of photographs, a list of photographs to a patient. They will fire only when I show a given photograph the very first time. The second time the patient sees the same photograph again, the neuron will no longer fire. So that's memory, right? Because that means something must have changed.
Yeah.
And that neuron signals the result of a novelty detection process. And we have studied those neurons in great detail and have shown that indeed they seem to be triggering the formation of memory when they fire.
Oh, how interesting. And so they are, let's bring that back to what you said earlier about forming a protein that stores that memory. So is that the moment when that protein is formed?
So the protein, it's the result of that. So it takes a while for a memory to stabilize. So the how these proteins, the synaptic proteins, are inserted and formed, it's thought to go on for several hours or days after you had that experience. But it's certainly the formation has been initiated at that point of time. Yes.
So that leads me to some questions that are apparently very popular. I did some research and studied what are the most frequently asked questions about memory. And one of those kind of ties back into what you just described there. So people are asking, there was the movie Total Recall, where there were memories implanted in various actors, and you had an experience as if it was real. So is the ability to insert those proteins or somehow stimulate them to create that protein, is that even possible? Is that something we might see in the future?
Not anytime soon. I mean, conceptually, in principle, we could certainly imagine doing that. But at the moment, we don't know the answer to some very simple questions. For example, the first time I saw you today when you popped up on my screen, how many of my synapses changed? And where are those? We don't know the answer to that question. Was it, 5? Was it 1,000,000? Was it 1,000,000,000? So to implant the memory, we would really have to redo that process, right? That happened in my brain when you popped up on my screen. So that the basic biology of which synapses are modified to create a certain memory really doesn't exist. There's much fundamental research that still needs to be done. So in principle, we could imagine doing that. In practice, we would have no idea how to do it.
Understood. Well, I guess that's kind of good to know, actually, that
yes.
that type of brain control is not yet possible. What another one of these highly popular questions is, why is it that I can sing along with this song that I haven't heard in years?
Yes, that's really a very common experience. And this is similar to the experience of if you learn to ride a bike, let's say, or swim as a kid, and then you don't do it for 20 years, you can still ride the bike and swim. So it just points to the fact that such memories, these are called procedural memories, so sort of detailed motor memories, sequences of motor movements. Once they are formed, they are lifelong. They will never disappear, and you may not know that they exist. It's not what's called a declarative memory. You can't talk about it, but once those sequences get triggered, they are there. They are lifelong memories, and they are very strong.
Fascinating. And the last of these highly popular questions is, why is it that dreams are so vivid and yet so difficult to remember?
Yeah, that's an excellent question, and it points to what we discussed earlier, which is the hippocampus. One of the remarkable things is that the hippocampus is inactive in the sense that it's not forming new memories, and that's why you don't remember it. There can be other situations like this also in real life. If for some reason the hippocampus is temporarily not making new memories, and that's the case while you dream, then it could be very vivid, but you will not remember it. As for example, during a traumatic event, it may have been super vivid, but it will not form a memory. So, and that's deliberate because you probably don't want to remember all of those streams.
Yeah, maybe not. So what does that tell us about the Alzheimer's experience? Is my hippocampus no longer storing and functioning correctly? Is that where Alzheimer's comes from?
Yeah, it depends on the stage, but yes, the hippocampus is, for reasons that are poorly understood, one of the brain areas that is most sensitive to damage. So it's often the brain area that is damaged first, and that is very much the case in Alzheimer's. And that's why this is usually the first symptom that we notice. It's our memory. And of course, that also degrades just as a function of healthy aging. But if that accelerates, then that's usually indication that the hippocampus is being impacted by disease. So that's very much the reason, yes.
Is there anything I can do to support my hippocampus?
Yes, all the things we talked about. So the hippocampus, as any other brain area, it's an area that you want to use as often as possible. You want to practice it. The hippocampus is remarkably also one of the very few areas in the brain that can grow in size as a function of use. There's this remarkable finding that I'm sure you have heard about the London taxi drivers who have to, if you want to get a license to be a taxi driver in London, you have to pass this incredibly difficult exam where you have to recall all the streets and all the directions, all the places. And a number of studies have been done, and those individuals have bigger hippocampi. And it's thought that their hippocampi grow in size because they use them so much.
So I am fascinated. I love to ask guests this question. So you have done a great deal of study in a very narrow area. What has that taught you? What lessons do you now follow to support your memory? Are there any techniques, tricks, tips that you can share that you put to use in your life?
That's a good question. Yeah, I would say so. I mean, I think the one thing that that has become very vivid to me is just how little we actually remember. Like if we study how detailed are our memories really, like, if I ask you, imagine what the face of your mother looks like. How much detail can you really bring to mind? It just has become very clear to me that this is really a precious ability that we are able to reconstruct memories or regenerate memories like that. But also how limited it is. We really actually don't remember that much. We have to really be humble about what we think, what we remember, because many of the things, if you really do tests, we just make them up, basically. Memory is often what we call reconstructive. Yeah, it's good to be aware of that. But yeah, I've become very cognizant of novelty. Really, I try to notice when something is novel, and I think that really makes me appreciate life more.
So more novelty in all areas of my life supports my memory and keeps my memory fresh, doing new things, new experiences.
That's correct.
That's a great excuse for getting out there and enjoying the world in a whole new way. I think I'll have to follow your advice.
Yes, I would recommend that. And certainly after the pandemic, that's a healthy thing to do.
Oh, I'm so on board with that. Thank you. That's Dr. Ueli Rutishauser. He is the founder of the Rutishauser Lab at Cedars-Sinai Hospital. Thank you for an interesting conversation about memory.
Thank you so much for having me. It was an honor.